what we're doing is very different. We don't know of a drug like us that's able to, in all the animal models, actually repair nerve damage, and not just repair, but repair it significantly. Every time we try it in the many models, from spinal cord to peripheral nerve to stroke, we see unprecedented repair, and it happens quick, and repair of many functions, from sensory to motion to cognitive. Of course, the forward-looking statements, and that's always important, but because I'm going to be sharing some good timelines here. So quickly, I am co-founder of the company and executive chairman, responsible in the finance side of things since the beginning. Recently, I took over as interim CEO as well, and Adam Rogers joined as interim president. And Adam actually his group invested $50 million US last year. So glad to have a large shareholder helping drive this company forward. We do have one of the world's largest search companies out there looking for a CEO. We've got a great short list that we're interviewing, and excited about making that addition in the next short while. And so hopefully we can update as we go. Also put up here, Dan Michael, very important, especially this year, as we expect to get our product into the clinic and administer to spinal cord patients even by mid-year. So Dan is designing the trials, and it's a really important statement that he made in joining us, because he was a practicing neurologist for many years, and then he joined the dark side of the companies, and but he worked his way up, starting Serono, Biogen, and his last position was really global head of therapeutics for neurology at NGEN, which is known as sort of the most successful biotech in history. So he was in a very cushy job, and but he actually left that for us because he saw the science and he wanted to make change. He wanted to do something really important, very positive about not just spinal cord, but MS, where he has a great experience, and even Alzheimer's, and pushing all those products. So he's going to be a key person for us for the next few years. So a lot of you know our story, obviously, so I'm going to just try and hit the highlights, but to reinforce, what we're doing is very different. We don't know of a drug like us that's able to, in all the animal models, actually repair nerve damage, and not just repair, but repair it significantly. Every time we try it in the many models, from spinal cord to peripheral nerve to stroke, we see unprecedented repair, and it happens quick, and repair of many functions, from sensory to motion to cognitive. And that is truly important. We at JP Morgan, it was noted, we arguably have one of the best data sets ever for a company of our size, and it's all on the web. But because we're repairing that, we have the opportunity to go after great, large markets where there is nothing repairing. They're treating the disease, treating the symptoms, but not repairing the damage. And that's truly important in a number of ways. And spinal cord is a great example, and that's the one we're driving at first, is that our clinical trial we're designing is 9 months in length. And usually a number of phase 2 spinal cord trials have actually failed over in actually completing, because they can't actually enroll the patients. What happens, a lot of them are neuroprotective, and you have to administer the drug to the site of injury within the first day, which is near impossible to get a patient to the right hospital within that day, get consent, while they're emotionally and physically dealing with potentially being paralyzed. In our case, we're going to be studying chronic patients, even up to 10 years post-injury, which allows us to enroll quickly. It's obviously we're the biggest hope in spinal cord injury, so there's a long lineup of people waiting to enter the trials. But what that also does allow for very quick, low-cost trials that read out quickly. And that is extremely unique. And we keep pressing that and wowing the investment bankers. Even in Alzheimer's, you're used to, a 2-3 year trial, even in phase 2, that's $100 million. But you'll see we're designing one that'll read out in a year, cost more like $20 million. And again, because we're repairing, you'll see a difference soon or you won't see it. And so it's not a matter of monitoring over years to see if there's a slight change. And that's truly revolutionary. It's a huge positive for investors. And that had a great reaction. So again, this is the one science slide. And again, we don't fix a disease. We don't treat a disease. What our drug does is go in the body and actually take the brakes off of the inhibition of repair, because the body knows since birth how to grow and how to repair itself. What happens when you damage the nervous system, whether that's from a spinal cord injury or even Alzheimer's, scars form and a certain protein is produced, and that's all has a purpose. It's there to protect from further damage, lock down the site, or maintain what's left. But what it's ended up doing is actually block and repair. And so again, we don't tell the body to repair a specific way. We just stop that inhibition. So we take the brakes off the inhibition and allow it to fix itself. And that is truly important. And you'll see, there's a number of many ways the body is able to repair itself. And we don't select it. The body selects depending on the condition. So for spinal cord injury, there's a lot of plasticity and some regeneration. We see some little bit of remyelination. MS, lots of remyelination, some plasticity. Stroke, we saw lots of plasticity, well-documented plasticity. And uniquely, the first time possibly ever observed, we saw stem cell promotion and migration through the scar and into the site of the injury, which is, we don't understand exactly what role they all play, but when it combined, the body knows how to fix itself, and you'll see just unprecedented repair. And here we are, an update is on our phase 1. We just started dosing patients in the last week in our very final cohort. And we did have a partial clinical hold. There was a reproductive toxicity issue in our first batch of drugs and testing years ago. And it's a legacy issue, and the FDA noted it. What happened is we couldn't find it again. We created new batches of drug. We stressed it every way, much longer studies, higher dosing, couldn't find the toxicity again. And in some ways that's good, some ways bad, because your best case would be to find the toxicity and then show that it went away and everything's good. But when you can't find it, you have to try to explain that first one. What we got out of the FDA recently was a letter that allowed us to proceed at a certain level. So they said, well, if there is some toxicity, there's a safety factor based on your highest animal toxicity, you're allowed to go to that level, that cap. Fortunately, that cap is the level that we're testing in our phase 1 patients now. And it's the level we were planning on taking into our phase 2. So for us, practically speaking, it was actually a go-ahead signal. Parallel, we'll be withdrawing the protocol likely with the FDA and hopefully coming off hold in the next short while. But operationally, we are now started dosing the bridging cohorts of the males and premenopausal women. Postmenopausal women went through flying colors. The drug was very well tolerated at a high dose. It was circulating the system, very long half-life, so it performed extremely well with no issues. And that was even up to even higher dose. So we're quite positive we'll get through this in the next short while. It's a 14-day dosing. We have all the patients enrolled, so we'll be starting administration shortly and moving forward. So that is hugely important to us after having dealt with this unfortunate, partial clinical hold issue for the last couple of years. it was great relief and excitement. We get to move ahead. And here's why we want to get into our spinal cord trial. I want to remind you again, the incredible recovery of these animals. And it's you'll see here visually, but even when we actually chart it, the recovery is not just unprecedented. There's nothing coming even close. In our last study that was done is independently in Germany on acute rodent population. The almost like 6 out of 10, and this was a severe injury model, 6 out of 10 recovered and were essentially up and running. And it's hard to find another study where one was even up and running. They're happy if a few get mild recovery. So totally unprecedented. Just to give you an example here. Again, on the left, this is an injured animal. You can just see the tails being dragged. In the back feet, they try to pedal a bit, but there's no weight bearing. Essentially, they're propelled on their front feet the whole time. And on the right is a treated animal, and you can see, and this is representative of the responders. The tail's up and moving, hind legs are fully functional, and this is truly remarkable. And it's interesting with the investment bankers last week that have seen lots of rodent studies over the time that they're still wowed by this amount of recovery. Another point, don't want to do too much data, but this came out late last year. And again, the big key word is the first one, chronic. This was injured animals, but we waited 3 months before administering our drug. And you can see at that point there was recovery, but at 3 months, the placebo animals flatlined. And you can see immediately with our drug, essentially, there was repair and strong repair. And also interesting, it recovered even after we stopped administering the drug. And so it shows that this, basically we're taking those brakes off, and those brakes, remain off. And when you look at this population with spinal cord, we believe that fix is permanent. Those connections are made, those new neurons are always there, so the injury won't reoccur. And this bodes really well. And then, so this is, again, the first time we've found anybody even attempting chronic, let alone having positive chronic data. And you'll see here how that's important. It's wildly important. It allows us in our phase 2 that we hope to start even as early as mid-year, 2 populations, a chronic population, which is 1 to 10 years post-injury, 10 injured patients and 10 placebo, and then a subacute, which is 10 to 49 days, or 1 to 7 weeks post-injury. So again, we don't have to go through that very difficult period of trying to enroll patients in the first day or two. It allows us to go, we don't have to go to the acute ward of the hospital to get our study done. So this will enroll quickly. We're doing it a single site, Shirley Ryan Ability Lab, Chicago, arguably the largest, most advanced spinal cord injury treatment center in the world, if not just US. Very high throughput, latest technology, best physio, very intensive. And so we're doing there as single site as well, because our primary endpoint is electrophysiology. Essentially, one of the electrical leads put in the top of the head and one in the toe. And with our drug, we expect to show increased, current through the body. That actually there is some electrical connection happening. So that's the primary. The second endpoint we'll do every, pretty much every other functional test known to man, motor. There's a good number of ways. There's bladder control, sensory. And so very intensive study, again, hoping to start this year with the chronic population, which will be fully enrolled within a month or two. Readout could be as early as first quarter of next year. and possibly earlier. If this does bring back, strong functional recovery, a drug like this in a trial, generally they are unblinded if there's, one study. And again, in chronic population, spinal cord, why it's important, after 15 months, maybe 18, there really is no return of function. You're pretty flatlined in your recovery. And so if our subjects, our patients actually have repair, we'll know where it's from. And so extremely excited. Now, what this does, I guess, again, a very low cost, $10 million study to get this done here in a short time frame. And this is very unique. And so this ability to add a chronic cohort to our trial adds a tremendous amount of opportunity. And we believe we may have been the first company to seriously do a spreadsheet, an NPV, on a drug that could actually access the chronic marketplace. It's very important. So there's 18,000 Americans that suffer spinal cord injury every year. About 300,000, though, are actually continuing to suffer and exist with an injury. And you can double that globally. But what this does, if there's success in chronic, change from a slower adoption into a market, which would be more like a rare disease model, still very successful in the acute, but it changes into what could be the first few years of sort of rocket fuel opportunity, where this great marketplace is looking closely at our drug and weighing the opportunity to take it. So we also, because of the demand of the cost of 1 to 5 years per injured person over lifetime care, there's a large ongoing cost. Therefore, there's ability to charge a price similar to a rare disease model. We have a market study that says we can charge up to $300,000. But even if you were able to charge $200,000 for a drug, and our advisory boards believe is fair price given the need, you're looking at a potential multi-billion-dollar revenue market. So it really changes the spinal cord injury opportunity on its ear. And historically, there's been a lot of failures in spinal cord, but none have had this incredible amount of supporting data early on. And none of them have attacked this opportunity from this angle. And so the reaction to this slide again at JP Morgan was really surprising and warming. They're seeing this isn't some little niche market. Now, we still have great reason to pursue the other markets. There's a number of other indications, as I mentioned, we have data on. These are our two priority indications, Alzheimer's and MS. Again, you look at that for phase 2 readouts in a year, very inexpensive. And Dan Michael, our CMO, has designed the studies. What we're going to do, though, is we're setting this up, continue to do some work, but really leaving it to the new CEO to figure out when and how to slot these into the clinical program. The one quick slide again to show you the strength. This is our recent news on stroke, and so there's one drug approved for treating strokes, and it's called TPA. But it's a drug that you have to give in the first 4 or 5 hours. And what it does is, if you've had a blockage in your brain causing the stroke, this will break it down a little bit and potentially reduce injury. So, except there's a number of patients that actually have a bleeding stroke. And so if you give this drug, you're going to bleed more and could die, which requires anybody taking drug to actually get like CT scan, MRI, some kind of a scan to find out what stroke they're having. So you have to get to a hospital, get scanned, and take the drug within the first 4 or 5 hours, or it doesn't help. So only 5 to 8% get this drug, and it still doesn't do much more than reduce some of the damage, in a very few people. So what we did a study, and we induced a stroke in the rodent models, and then we administered drug 1 day after the stroke and 7 days after the stroke. And in both cases, we saw a tremendous amount of repair. And so this is, again, truly unprecedented recovery and repair in an animal model. And we're trying to figure out where this fits. And again, the new CEO will have to figure out where this fits into our lineup. But what is also interesting, there's a quick video I'd like to share. And this is a rodent model. The animals have a stroke induced. And you see they're released, and they're trained to go down to the hole up on the upper left by the red mark. That's the exit. And you see a healthy animal that's trained knows how to exit that table within 10, 15 seconds. Then you induce a stroke, and this is placebo treated, so no drug. But they have a stroke induced, and they lose a lot of their cognitive function, their memory loss. And so it struggles around, goes around, and then finally, a minute and a half later, ends up going down its hole. And it's more by trial and error rather than remembering. And then this is an animal that actually was administered with our drug. And you'll see it didn't do it in 15 seconds, but you'll see it actually was able to do it in incredible fast time compared to the animal that was just received a placebo. And so again, great success, and importantly, repair of a cognitive function of memory. So what this, it does provide some information of plasticity in the brain, and we'll have to see if that imports over to Alzheimer's. So to wrap up, again, we have, well, we found a very respectable market cap of the company here. We always want to do better ourselves and keep fighting, get the stock up, but relative to companies at our stage in this market and the biotech world, we're doing very respectably. We will probably look at some point for top-up financing, but likely wait for our CEO to come on board, and I think that'll add a lot of stimulus to the shares and our ability to raise money. And so we do now have $20 million roughly in cash. We're well positioned in this market and not in any great need or urgency. So to wrap up here, lots of good value drivers. It's been a long road to work through this clinical hold issue, but that's wrapping up shortly. We hope to have that clinical hold removed completely. But frankly, it's allowing us to move forward and plan to execute and initiate a spinal cord injury phase 2 trial, even by mid-year. We're working on some other important disease models that could read out this year as well to provide good evidence of other indications. And we're quite late stage on a couple of grants as well, getting good support from organizations, both spinal cord and U.S. Department of Defense, which is highly interested in spinal cord and traumatic brain injury. And as well, we're looking forward to getting a new CEO on board in the next short while. And having that in place in this program, we're quite optimistic that we can look towards an uplisting even to NASDAQ, hopefully even this year. And that was the reaction we're getting from JP Morgan, that there is great support for us to move this program forward. If we're getting towards phase 2 and into phase 2, they expect that we'll be able to find the right bankers and investors that support this up onto the NASDAQ.
Okay, sorry. No, so thank you so much for that summary, Bill. It was great, and I think it really highlights the opportunity that the company is in front of. I have a couple of questions there. So if you're successful in your current clinical program, the phase 1 and the phase 2 in humans, which is clearly an exciting point, but assuming that if you are getting some significant improvement in those patients that are without hope, essentially, there is a high likelihood that you will get an orphan drug approval that you could start to, or on a compassionate use, right, to sell. Yeah.
Yeah, no, that's a good point. There's a number of avenues, and we do expect to proceed much faster than usual. We did have orphan status in Europe. We didn't get orphan status in the US, though, which was, in a way, to me, it was almost worth the news release, because one of their arguments was that they believe this may be able to treat chronic patients, which puts it much larger market patient population than fits with orphan, which was fantastic news for me. And orphan status isn't so important. What is important is to find the support to have an accelerated or expedited review. Truly, if we bring strong repair, and again, what's the hurdle isn't necessarily someone getting out of their wheelchair and walking again. If you could return bladder control, I mean, that is a home run for many people. It's almost a death sentence for a lot of people, the amount of bladder infections that they get. So return of their sexual function, bowel, there's so many different issues they deal with that, for us, the hurdle to an accelerated approval is fairly low.
Would you say that the following summary may be kind of giving the picture of what is happening? So when somebody gets an injury, there is a whole lot of biological processes that start to try to heal, but potentially the most important thing is to stop the damage. And so the research behind your technology is that you were able to identify some of those factors, some of those proteins probably, that were inducing, or where their main job was to stop the damage. And when you now inhibit them, meaning you stop them from doing their job actually, then you allow that natural repair process to actually continue.
Yeah, well, yeah, there's a little tweak in there. And so this is, lifelong work of Dr. Jerry Silver at Case Western, and in spinal cord injury, neural research, he's very godlike. And so he was the one that really changed the understanding of nervous system damage, on its head. Previously, they thought it was more of a mechanical block, something like that. Nerves are just dead. But he was the one that did the work and just thought outside the box and discovered that, when the nervous system has damage, there's this one specific protein, proteoglycan, that is in the scar to as you mentioned, lock down the site, stop further damage. And so he was one that identified that proteoglycan as also the one most responsible for preventing repair. Our drug, though, importantly allows the scar to keep functioning and doing what it's there to do. So all that protective function is still there. All we're doing is preventing it from inhibiting the repair.
Right.
And so that's very important. There's other enzymes and such that can break down the scar to bring back function, but you really want to leave that scar intact. And so that was the big discovery, is finding this peptide that allowed that natural Darwinian protective function to maintain itself, yet stop the inhibition.
Yeah, well, fantastic. And the second thing to highlight, which you already have done, but to do it another time, is like, this is not only something that could allow for nerves to regenerate and repair following an immediate injury. It has probably applicability to other diseases like multiple sclerosis and also to Alzheimer's, which would allow the nerves that are essentially breaking down to continue to work the way that they are supposed to be working, and therefore alleviate the symptoms and even potentially reverse the symptoms of those diseases.
Yeah. Oh yeah, no. Theoretically, you could add ALS.
Yeah.
You could add epilepsy into that. Both, damage the nervous system that needs repairing. Traumatic brain injury. Yeah. So there's anything really. This proteoglycan appears anywhere there's damage to the nervous system, and that's stopping repair. So when that's present, we can counter that effect and bring back the repair.
Well, thank you for your time, Bill, and thank you for the introduction, and we will clearly update as we go forward.
Looking forward to it. Thanks for the opportunity.